Good morning to one and all. On the occasion of National Science Day 2024, with the theme Indigenous Technologies for Vikasit Bharat, I am going to discuss the topic entitled on Biotechnological Innovations in Gastrointestinal Infection Diagnosis. Hi, I am Dr. Venkateswara Prasad. I have done MSc Biotechnology, followed by PhD in Biotechnology. Presently working as an assistant professor in Crescent College, Mandalur, Chennai. So my area of expertise is microbial genetics, cancer biology, diphtheria, diarrheal diseases, and non-communicable diseases like identity deficiency disorder, fluorosis, and anemia. Presently, I completed 4 extramural projects that is comes under ICMR, and one intermural project is going on. It is funded by the seed grant fund from Crescent College. And along with that, I have submitted novel genomic sequences from various research works that was submitted to GenBank, NCBI, National Center for Biotechnology Information. This is about my research profile. So coming to the topic, the gastrointestinal infections. So what are these infections? These infections are caused by bacteria, viral and fungal pathogens. So actually this gastroenteritis, the word is applied to syndromes of diarrhea and vomiting. So what is diarrhea? Diarrhea is commonly defined as 3 or more loose stools in a 24-hour period. If you are not treating properly or in early stages, it will lead to a severe acute conditions. It may carry over 14 days. It may prolong to leads to 14 days infections. So it leads to severe organ failure, and it may lead to death also. So what are the causating agents? The causating agents are bacteria like cholera, E. coli, salmonella, Shigella, viruses like enterovirus, rotavirus, hepatitis A, and parasites, and and protozoan parasites, example Giardia lamblia. And here we have to distinguish two words. Those are food infection, food toxification. So food infection is causing agents, or these bacterial agents are like Salmonella, Shigella. And food intoxication means the causating agents are Staphylococcus aureus, Bacillus cereus, and Clostridium perfringens, and Clostridium botulinum. Coming to disease burden of the gastrointestinal disorders around the world. So the global burden of disease study found that diarrhea is the 9th leading cause of death globally in 2015, and was responsible for 8.6% of the death among children younger than 5 years. The greatest impact of diarrheal illness is among children younger than 5 years in low and middle income countries like India and other South Africa. That things is the second common cause of death beyond the neonatal period. Approximately 179 million cases of acute gastroenteritis occur in the United States of each year. Coming to mortality rate due to gastrointestinal disorders, according to Encheng Wang 2023 data, the leading cause of death among digestive disease in 244 countries and territories in 2019. So the main cause of death is enteric infections. So it is around 57. This slide is showing various colors regarding that the mortality data. So highest is cirrhosis and other chronic liver disease. It is 67. So after that, colon rectum cancer, and after that enteric infections. That is the reason why this digestive disease leads to mortality rate when compared to other diseases. Coming to early and accurate diagnosis of gastrointestinal infections. So why you want to diagnose early and accurate diagnosis required for treatment? What is the need of the hour? So here I am going to discuss in 6 points. First point is timely treatment. So it allows for prompt initiation of appropriate treatment. In many cases, timely administration of antibiotics, antivirals, or whatever antiparasitic medication can significantly reduce the severity of the infection. That is the first point. And second point is prevention of complications. Complications means the gastrointestinal infections. It may lead to very severe conditions like organ failure and electrolyte imbalance and dehydration. It leads to severe complications. So we have to stop. For that reason, we have to diagnose properly in early stages. So the third point is reduced transmission. It is not only like preventing the complications, stopping the complications, and we are going to reduce the transmission. That means from person to person. If the infection disease, it will transmit from person to person in a community. It will lead to outbreaks. That is also one of the important point. Coming to public health impact. So early diagnosis plays a vital role in public health surveillance and response. Identifying address key outbreaks quickly can prevent the further spread of infections within communities or across the regions. So another important point is minimization of the antibiotic use. Nowadays, the people are using without any prescription the antibiotics. So that is a very dangerous step in coming days. So accurate diagnosis helps in prescribing the appropriate antibacterial agents. So last point is early diagnosis can lead to more cost-effective healthcare. Timely treatment always less sources intensive than managing advanced or complicated cases. It can also reduce the economic burden of the both individuals and healthcare system. Coming to transition to biotechnological innovations, because of why you want to transition to biotechnological innovations, because of limitations of old traditional methods. So old traditional methods may not give proper diagnosis information, proper diagnostic results in terms of speed and efficiency, precision and specificity, point of care diagnostics. It may involve it in molecular diagnostics. All these things we can overcome by using these biotechnological innovations. So it will be useful in understanding the microbiome and research and development focus. Coming to the biotechnological innovations or advances in the field of biotechnology. So here I am going to discuss three innovations it comes under in biotechnology. One is a polymerase chain reaction. It made a revolution in biotechnology history. And second is next generation sequencing. And third is CRISPR based technology for target detection. So coming to one of the important innovation comes under biotechnological innovations, that is PCR. PCR stands for polymerase chain reaction. It is a technique. It is used to amplify your gene of interest wherever you want from the source of your genomic sample, DNA sample. So this PCR is invented by Karamulis in 1983. It's very it will be useful in diagnostic purposes. It is not only useful in diagnostic, will be useful for forensic application, agricultural, etc., etc. So this PCR, it consists of three important steps. What are those? These are denaturation, annealing, and extension. So this denaturation means it is a double stranded DNA. It is unzipped into single strands. Annealing means it is primers base pairs with the target DNA sequence. So especially related to your annealing temperature. Annealing means it is a binding temperature, the primer with the template sequence. So coming to the third step, extension. So extension means tag DNA polymerase extend the primer and synthesize the new strands of DNA. To the PCR components, what are the components, important components are involved in PCR reaction, thermal cycle reaction? That is, PCR reaction practically it will require components. What are those components are template DNA, DNA primer, it's also called oligonucleotide chains, and tag DNA polymerase and nucleotides. These are the important components involved in the PCR reaction. After mixing the PCR components DNA template, deoxynucleotide triphosphates, tag DNA polymerase, and then buffer. After mixing all these components, we are placing the PCR tube in the thermal cycler machine. Here in this slide, I am showing that PCR machine. Those are the three steps. Those are denaturing, annealing, and elongating the temperatures approximately 30 times. After repeating of 30 times of these three steps, denaturing, annealing, and elongation, the PCR reaction is going to complete, and it will give number of copies, and it is like this. I'm showing this slide. That is a number of copies of your gene of interest, gene of your DNA sequences. This will be useful for your characterization of the genes and cloning and further studies also. Coming to the applications of PCR. So this is the main technique used in biotechnology innovations. It will be useful in diagnostic medicines. Example: detection of infectious agents, bacteria, viruses, parasites. It is also used in genetic screening for hereditary disease like abnormality of like genetic hereditary sickle cell anemia or hemophilia or thalassemia or etc etc. It is not only useful for screening for hereditary disease. It will be also useful for prenatal diagnosis to find out the like like fetal abnormalities also. So it is also useful for to find out the viral load for hepatitis A and hepatitis B viruses also. And not only it is using in diagnostic medicine, it will be useful in research and biotechnology, especially cloning and sequencing of DNA, gene expression analysis, and genetic engineering and manipulation. And it can also be useful to detect of microbial contaminants present in water and soil, monitoring and biodiversity and ecosystem health. It is also useful in study of phylogenetical relationships between the organisms. Whatever we have isolated and we are doing further characterization, it will be useful in characterizing of the microorganisms also. Second important technique used in biotechnology innovations for detection of pathogens. So CRISPR-based technologies for target detection of pathogens. So CRISPR-Cas system. What is CRISPR? The clusteredly regulated interspaced short palindromic repeats. And CRISPR-associated proteins are part of the bacterial immune system. They can be harnessed for nucleic acid detection. So CRISPR, Cas12 and Cas13 detection. So when a target nucleic acid sequence is present, CRISPR-Cas12 and Cas13 systems become activated. Cas12 cleaves merely single-stranded DNA and Cas13 cleaves RNA. The activated system produces a detectable signal. And third important thing, Sherlock. Sherlock means specific high sensitivity enzymatic reporter unlocking. This system utilizes CRISPR-Cas13 to detect and cleave the RNA target sequence. The collateral cleavage of activity of Cas13 is harnessed to generate a detectable fluorescent or colorimetric signal. So it will be useful to detect the pathogenic sequences present in the sample. In this slide, we are showing two technologies. One is the detector technology and another one is Sherlock technology. In detector technology, we are using Cas12 for detecting the pathogenic sequences. So it will give fluorescence and it will give some signal. So based on the signals, you can identify the pathogens. In the similar way, here we are using a Sherlock technology, but in this technology, we are based on the targeting sequence of RNA, it will give signal. Here system is Cas13 will be used to identify the pathogenic RNA sequence. This is the difference between detector and Sherlock technology. Coming to advantages of CRISPR-based detection system technology. So one of the important advantages is high sensitivity and high specificity. So by using this technology, you can achieve high sensitivity and specificity for the targeted sequences present in your clinical sample. And it is the second advantage is rapid results. It will give very rapid results, making them suitable for point of care applications. And third advantage is multiplexing capability. The system is this CRISPR system can be engineered for multiplexing detection, allowing the simultaneous detection of multiple targets. So the clinical samples may contaminate with the it will contain some other pathogens also. It will be detected by using this multiplexing capabilities. And another advantage is adaptability. So this CRISPR technologies are versatile and can be adapted for the detections of various nucleic acid targets, including those associated with the infectious agents. And one of the last important advantages potential for point of care applications. Ongoing efforts are focused on developing portable and user-friendly CRISPR diagnostic devices for use in diverse settings, including resource-limited environments. Coming to lost, the third technology, that is the next generation sequencing technology. This technology is a powerful high throughput technology that has transformed in the field of genomics. It allows for the simultaneous sequence of millions to billions of DNA fragments, providing comprehensive insights into the genomic makeup of organism. NGS, next generation sequencing, has numerous applications including whole genome sequencing, RNA sequencing, and metagenomics. Here is an overview of how NGS is utilized for comprehensive analysis. First one is whole genomic sequencing. NGS whole genome sequencing involves sequencing of the entire genome of organism, providing comprehensive view of genetic content. So it is useful in genome variation analysis, especially identifying single nucleotide polymorphism, insertions, deletions, and structural variations present in samples. It is also be utilized in genome assembly, reconstruction of complete genomes, especially useful for non-model organisms or those with complex genomes. This slide is showing the methodology of NGS technology, how the clinical samples blood, stools, urine, sputum sample will be utilized for genome sequencing of genome sequencing by using NGS technology. After this, it will lead to second step. It will go to the next step called characterization of pathogen. Then it will go to the next step of development of biomarkers and biosensors. Ultimately leads to application and rapid identification of the pathogen. This is the one way of developing the rapid identification of pathogen. And another way is if you can directly isolate the DNA for PCR based rapid detection of the pathogen. That is also will be possible to identify the specific particular pathogen by using this technology. Coming to metagenomics, metagenomics involves sequencing the genetic material directly from environmental samples containing the mixture of organisms. So this metagenomics will be utilized for profiling the diversity and abundance of microorganisms in the complex ecosystems, and it also be utilized for identifying microbial pathogens in clinical and environmental samples. So here one of the another technique is also there. That technique is chip sequence. That means chip means chromatin immunoprecipitant sequencing. This combines with the chromatin with the NGS technology to identify DNA binding sites of the specific protein. Biotechnological innovations have significantly advanced in the field of gastrointestinal infection diagnosis, but several challenges persist. Here are some challenges and potential future directions. The first one is cost and accessibility. So some advanced biotechnology methods can be expensive and may not be readily accessible, particularly in resource and limited settings. So solution is continued efforts to develop cost-effective technologies and promote their accessibility globally. And second challenging point is the implementation of certain biotechnological methods may require specialized infrastructure and skilled personnel. For this challenge, solution is training programs and infrastructure development to enhance global capabilities in using advancing diagnostic technologies. And third point is sample complexity. So sample complexity, the complexity of clinical samples such as stool or blood can present various other contaminants in extracting and analyzing target DNA and RNA. So for this challenge, solution is ongoing research to improve sample preparation methods and increase the robustness of diagnostic assays. And fourth challenge is standardization and quality control. Ensuring consistent, reliable results across the different laboratories and assays can be challenging. So for this point, development of standardized protocols, quality control measures, and proficiency testing programs, it will be useful to overcome this challenge. So integration with the healthcare system. The challenge is integrating biotechnology innovations into existing healthcare system may require changes in workflows and infrastructure. The solution for this point solution is collaborative efforts between technology developers and healthcare providers and policymakers to streamline the integration. So future directions. It will be useful in point of care technologies, continued development of user-friendly, rapid and portable point of care diagnostic technologies for immediate on-site testing. It will be mainly useful for outbreak or recent outbreaks uh, calamities. So, and multi-omics approaches, integration of multiple omics data, genomics, transcriptomics, and proteomics for comprehensive understanding of gastrointestinal infections and personalized treatment strategies. It will be useful in establishment of global surveillance networks that leverage biotechnological innovations to monitor and respond to emerging gastrointestinal infections. It will also be useful useful in microbiome therapeutics. There is a future direction of this biotechnology innovations. Utilization of microbiome data for the development of targeted therapeutics, such as personalized probiotics, to restore and maintain gut health, and environmental monitoring. So, if you implement of biotechnological methods for monitoring environmental factors that contribute to the spread of gastrointestinal infections, such as water quality assessment and other things. So it will be useful in public health education also, in terms of increased efforts in public health education to raise awareness about the benefits of early diagnosis and the role of biotechnological innovations and the importance of preventive measures. It will be useful to prevent the outbreaks in community in rural settings.